Welcome back for more AP Biology. Today we're going to deal with stem cells. This isn't a lecture that you really need to take notes about, even though they've talked about this, which is why it has the odd numeration at the top. But sometimes it's nice to have a little bit of a background. So we're going to deal with the environment and what type of cell you get. And this actually is something that the College Board asks about. So in order for us to get cells, what we ended up, or a multicellular organism, step one is we need to have some type of starting point. If we're assuming that we have sexual reproduction, what we need to have are two events that occur in fertilization. The first one is called plasmogamy, which is where the cytoplasms fuse. The second step is called karyogamy, which is where the nuclei fuse. We assume that both of these happen at the same time, but they're actually organisms, a whole bunch of fungi, that don't follow this pattern, which is why it's worth pointing out. So when I look at fertilization, in us, in animals, it's clear what's going on because we happen to have a rather large egg and a rather small sperm. The two run into each other. Hooray, we get plasmogamy, nuclei fuse, we have karyogamy. The catch is, in not all organisms is it this straightforward, because some cases we can't tell the difference between the sperm and the egg, so rather than saying sperm and egg, we'll say a plus and a minus. And you need to have a plus and a minus in order to fertilize, and if you have two pluses, it doesn't work, two minuses, it doesn't work. What ultimately happens is we get that plasmogamy, we get karyogamy, and that cell is then going to divide. When it divides, it starts out with one cell, and that's all it is. All of its contents, all of its surroundings are the same. The moment we divide, we have two cells. What we end up having are two different sets of environments. We have an environment where two membranes are touching. Then we have membranes on the outside. Why is that important? Because now we can have two different sets of signals, because the environment dictates signals. Signals can modify chromatin to open it up or tighten it up. Those signals also can go through signal transduction and express genes differently. So both cases we can get different genes expressed because we have different sets of environments. When I go from 2-cell to 4-cell, I am increasing the cell-cell contact and decreasing the outside influence. When I go to the 8-cell stage, we have a lot more cell-cell touch and not as much touching the outside. But the moment I get what we call the morula, or the early blast, or then we start going into the blastocysts, what we end up having are cells that are on the outside, and we have cells on the inside. The result is we have two completely distinct environments, and the result of that is we will have different genes being expressed. How can we tell? Because when we look at the blastocysts, these bottom two, we end up forming a cavity. Why would that cavity form? Because different cells are having different sets of genes expressed. The result is we are getting differentiation in our cells. If I keep looking at that, those cells will ultimately become other types of cells. We call those cells that can produce other types of cells stem cells. And there are varying degrees to which these exist. If I keep looking at that, the initial zygote, what that could do is make anything and everything. Ultimately, we got that cell, the blastocyst, that looked like it was a soccer ball with another thing chunked on the inside. That piece on the inside is ultimately will become you. Those types of cells are what we call totipotent stem cells. The name doesn't matter. And those ultimately will become one of three types of cells, what we call endoderm, mesoderm, and ectoderm. I can only get those stem cells traditionally from an embryo. I had to go in, wait till it goes through so many cell divisions, and then I can start plucking those cells out. And then I can, if I know the right environments, meaning I know which genes to turn on and which genes to turn off, ultimately I could make whatever I want. Because the endoderm, as you can see, can end up making, in this particular case, lung or pancreas. You can make more than that. Mesoderm can make heart muscle or red blood cells, and the ectoderm can make skin or neurons. That's traditionally how it would work. Well, what if I needed to replace some neurons? Well, old-fashioned way would be, let me get an embryo, wait till it gets to a certain stage of being a stem cell, pluck that stem cell out, grow it under certain conditions, I end up getting a neuron, hooray, and a hip hip, moving on with existence. The catch with that is you introduce a whole bunch of ethical issues And are there ways around it? Well, it turns out, if I know the switches that can turn that ectoderm line stem cell into a neuron, if I know the series of steps that are needed to do that, why can't I reverse engineer it? Or I can take a skin cell. If I know the series of enzymes and transcription factors that you add to turn that ectoderm line stem cell into the skin cell, what if I could flip the switches backwards? So I can reprogram a skin cell to become a stem cell, which I can then, using a different set of switches, turn that into a neuron. That process is referred to as induced stem cells or induced pluripotent stem cells, IPS or PSC. The I is a lowercase I. And that is something that we are capable of doing. Because if I know the order of the environmental factors that turn a stem cell into a particular type of cell, I got it. What can we do with these stem cells? The answer is all sorts of things. So again, iPSC, induced pluripotent stem cell, meaning I made a stem cell that can become all sorts of things. Well, why would we care about this? Well, it turns out, in class you end up reading an article about this, but the way that we made that happen is we modified stem cells. It used to be you had to know a series of different steps and you had to reverse backwards and then move forward, and you had to move cells between different types of environment and through different media. But it turns out there's a shortcut, as recently discovered. So what we can do is we can directly edit genes. We've known this. You did a lab on this. So we could use restriction enzymes. We chop up DNA. We can put sticky ends back together. Haha, we made a recombinant piece of DNA. That's not so bad. It's a protective mechanism that bacteria have. If we look at our protective mechanisms, we have either defense or we have immunity. Part of our immune response is we remember everything that has caused us harm. The way we can do that is we just need to keep some cells that remember floating around. If I look at a bacterium, they don't get that option. They can't make other memory cells because the one cell is all they get to have. So it turns out that they have a mechanism that we call CRISPR. And what CRISPR can do is take a cell that is invading, or a virus that invades, if you happen to be aware of it, what we can do is use this mechanism called CRISPR to chop up the invading DNA or RNA, doesn't matter, and we can have a big enough piece of it so it's incorporated into the genome. So in a sense, it's being remembered. What we can then do is express a gene that becomes a guide, something that we call Cas9, that if I ever get that virus to show up again, I can now say, hey, I know what you are because I have a piece of DNA, or RNA in this particular case, that matches your genetics. I know what you are, and it attacks it, chops it up. So it's a way of actually having a bacterial immune memory. Well, what's the point of this? I can use CRISPR to actually go into organisms and edit their genomics. I can edit their DNA in the living organism. All I have to do is give this system the correct target, and the result of that is I can change DNA. Why is this important? Because if I know what genes need to be turned on and which genes need to be turned off, I could probably use CRISPR to do those modifications for me. I don't need to do all these cells grown in all these media and moving them back and forth. I could just jump straight to the start. And that's useful for your article that you're going to be reading in class.